Hi guys, Miss Francis here. We left off describing Watson and Crick's model of DNA. And they described DNA as this double helix where you have alternating phosphates and sugars that form the backbone of DNA. How do I know whether I'm looking at the phosphate or whether I'm looking at the sugar? The sugar is connected to what are called your nitrogenous bases. These nitrogenous bases form what is called the rungs found in our DNA double helix. Do you notice how each rung has 3 rings? We know that a pyrimidine pairs up with a purine to give us our 3-ring rung. And we also know that there are base pair rules that Chargaff gave to us where T's and A's match up and G's and C's match up. We also know that the bond holding together those nitrogenous bases are relatively weak hydrogen bonds. And we know that between thymines and adenines, we'll have 2 hydrogen bonds. And between guanines and cytosines, we will have 3 hydrogen bonds. Now, not only did Watson and Crick propose this model for the structure of DNA, but Watson and Crick also published a second paper that explained how DNA replicated. And their proposal was that existing DNA strands serve as templates for new complementary strands. So they gave us what's called this semi-conservative model, and there are 3 different models that were actually proposed. Before we get to the models, let's take a look at the overall process of replication. This is the process that Watson and Crick proposed. So here we have our parent, Here we have our parent strand of DNA. Watson and Crick stated that the hydrogen bonds between these nitrogenous bases break and our parent strand separates into template strands. So the first step is the separation of the 2 DNA strands. Then the second step is that these parent strands serve as the template for the new strand, where nucleotides arrange themselves, where the nitrogenous bases within the nucleotides follow the base pair rules. Lastly, the nucleotides are connected to form the phosphate sugar backbone, and then each daughter molecule, so this is a daughter molecule of DNA and this is a daughter molecule of DNA. It consists of the parent strand, here's our parent strand, the dark blue, and 1 new strand, where the new strand is our light blue. Messelson and Stahl were 2 scientists that set out to find evidence to support Watson and Crick's proposed model of replication, or to find evidence against his model of replication, because at this time there are 3 different proposed models. So what Messelson and Stahl did was they took E. coli and they allowed E. coli to replicate in the presence of heavy nitrogen. When that bacteria replicated in the presence of that heavy nitrogen, the heavy nitrogen was incorporated into their DNA because DNA contains nitrogen in their nitrogenous bases. Using a centrifuge, which is a machine that separates a sample based on its density, the Messelson and Stahl took their sample and they spun it down. And when they spun it down, what Messelson and Stahl ended up with was 1 band at the bottom of their test tube indicating that the DNA contained this heavy nitrogen because the heavier substance went further down in the test tube. Their next step was to allow this E. coli to replicate in the presence of nitrogen-14. Nitrogen-14 is considered this light nitrogen. When the E. coli replicated in the presence of just the light nitrogen, it incorporated the light nitrogen into its DNA because DNA contains nitrogen in its nitrogenous bases. And now this time, when Meselson and Stahl spun their sample in the centrifuge, what they got was a band in the middle of their test tube, where this band contained both light N14 and heavy N15 nitrogen. Once again, Meselson and Stahl allowed the E. coli to replicate in a medium containing just this light nitrogen, spun down their sample, and what they ended up with was again a band in the middle of the test tube indicating that it contained both light and heavy nitrogen. But this time, they also ended up with a band at the top of the test tube, which means that that DNA only contained the light nitrogen. So now these are the results that Meselson and Stahl got when they did their experiment with DNA. The three proposed models explain the method for DNA replication. So using the information that Meselson and Stahl got from their experiment, they compared it to the results that they would get if each model for DNA replication held true. So the first model that was proposed, or one of the models, is the conservative model, which says that the helix, the double helix, doesn't separate. Instead, it remains intact. And instead, you get an identical copy of the first strand that is all new. The second model that was, or a different model that was proposed, was the dispersive model. And the dispersive model says, yeah, the strand separates where each daughter strand contains a mixture of old and new parts. So it would contain a mixture of N14 and N15. The model proposed by Watson and Crick was the semi-conservative model. And Watson and Crick said that the DNA unzips, the hydrogen bonds break, and the parent strand, the strand that was tagged with the N15, would serve as a template for the daughter strand. And in this case, the daughter strand was tagged with the light nitrogen, the N14. So let's follow along and see what happened when Meselson and Stahl followed the patterns that were proposed for the different types of, or different models of replication. So the DNA gets tagged with heavy nitrogen, right? This heavy nitrogen being N15. So here I've got my DNA, and it's going to get tagged with my heavy nitrogen. When I spin my sample in a centrifuge, I get one band at the bottom containing all of the heavy nitrogen. Yep, that matches Meselson and Stahl's experiment. Now, according to the conservative model, my DNA strand does not separate. Instead, it replicates itself. And now Meselson and Stahl exposed this strand to a medium containing only nitrogen 14. So if this were the case, if the conservative model were to be true, this new strand would contain only nitrogen 14. And when Meselson and Stahl would have spun this down, they would have gotten one band at the top of the test tube that contained only the nitrogen 14 DNA. And then they would have gotten one band at the bottom of the test tube that contained DNA tagged with just the nitrogen 15. However, this is not what happened when Meselson and Stahl did their experiment. So they knew that the conservative model wasn't plausible. Moving on, Meselson and Stahl looked at the dispersive model. Once again, the dispersive model, Meselson and Stahl, starts out with DNA that's tagged with heavy nitrogen. When spun in a centrifuge, this heavy nitrogen would form one band at the bottom of the test tube containing all heavy nitrogen. Check. That's what Messelson and Stahl saw in their parent cell. Now, according to the dispersive model, our DNA does separate. It does unzip along the hydrogen bonds, but these new strands contain a mixture of the parent strand, the N15, and the new strand, which would be the nitrogen 14, because Messelson and Stahl now grew their E. coli only in the presence of this light nitrogen. And if this were the case, Messelson and Stahl would have seen a band in the middle that contains the light nitrogen and also contained the heavy nitrogen. Well, guess what, guys? This is what Messelson and Stahl saw. So far, the dispersive model is looking pretty good. They enabled this E. coli to replicate one more time in a medium containing only nitrogen 14. And if the dispersive model were to be true, the parent strand would contain a mixture of the light nitrogen, this blue nitrogen, and the heavy nitrogen, which is the blue nitrogen, and it would contain also the light nitrogen, the pink nitrogen. Now, if this sample were to be spun down in a centrifuge, once again, Messelson and Stahl would have seen a band in the middle of the test tube containing equal amounts of the heavy nitrogen and the light nitrogen. Well, guess what? This is not what Messelson and Stahl saw. That's not what they saw. So they reasoned, okay, this can't be the proper model that explains the way DNA replicates. So now they looked at the semi-conservative model that was proposed by Watson and Crick. And Watson and Crick said that their original parent template that was tagged with the nitrogen 15, that when spun down would form one band at the bottom, separates. And now these parent strands that contain nitrogen 15, when grown in the presence of the lighter nitrogen, would be half light nitrogen and half heavy nitrogen, because this new strand would be paired up with the parental strand, the strands containing the nitrogen 15. And we would get a band in the middle of the test tube containing equal amounts of heavy and light nitrogen. This is what Messelson and Stahl saw. Lastly, Messelson and Stahl allowed their DNA to once again replicate in the presence of light nitrogen. So now we've got our 4 parental strands. 2 parental strands consist of heavy nitrogen, and 2 parental strands consist of the light nitrogen. So when the new strands form related to the template, do you see how I would have these 2 strands made up of entirely just light nitrogen, and when spun down, they would form a band at the top of the test tube, whereas these 2 strands are a mixture of the light and the heavy nitrogen and would form a band in the middle. And guess what, guys? This is what Messelson and Stahl saw. So they concluded that Watson and Crick's proposed model for DNA replication, the semi-conservative model, must be true. There are some practice questions here that you should take a look at and try to master.